Let's talk about the next part. So, what is the flu virus like on the inside? And so I think it's pretty neat. So, on the inside, you've got 8 of these RNA segments, and they're segments of negative sense RNA. So, it's just regular RNA, but it doesn't actually encode a gene, it encodes the reverse complement of a gene. So, everything is backwards, and all of the bases are the opposite of what they should be. So, like if it should say ATG, instead of that, it's going to say backwards TAC. So, yeah, tac, tac, tac. Each of these segments actually forms up into a structure that is called a panhandle. So the 5' and 3' ends of each segment will bind to each other. So that's what you can see over there at the left side of the model as kind of inside of the sphere for PB1, because PB1 is what's going to bind to this. So the panhandle is just the front and the back ends of the RNA joining together. And so the rest of the virus segment, imagine that big circular bit of RNA, that's the pan, and the handle is the bit where it's stuck together on either side. So there you go, it gives you a nice sense of it. And so PB1, PB2, and PA are the 3 parts of the polymerase. And so they are together going to do the job that the NSP12 polymerase did for coronavirus. And they're together going to do the job that the reverse transcriptase did for HIV. Now there's a function for each one of these, and that's pretty neat. And so this is another thing I'd like you to get good notes on. So the function for these is that PB1 is the actual polymerase. This is actually going to copy the RNA. PA is a cap-snatching enzyme. So what it does is it's going to find a messenger RNA from the cell, which is going to have a nice cap structure and a nice poly A tail, because they all have to. And it's going to grab onto this, and it's going to snip off the cap part plus the first couple of nucleotides, and then it's going to use this. It's going to pass this over to PB2, and so PB2 is like the middleman. This is our shady middleman, and PB2 is going to hand off this stolen cap to PB1. It's like fencing stolen jewelry through a pawn shop or something. Think of PA as the burglar, and the PB2 is like the pawn shop, and PB1 is the customer. It just wants uncomplicated RNA caps. So whereas the coronavirus, like I said, blue collar virus, going to go out and make its own darn cap with its own, not hands but proteins. Influenza is a nasty, dirty little thief of a virus, and it just grabs one of your messenger RNAs from the cell and beheads it, cuts off the cap part, after which your messenger RNA does not work and is useless and will break down. And it feeds it over to its polymerase, and it turns it into a new copy of influenza virus. So, yeah, that's pretty lousy, right? All right, so each of those 8 segments is going to be negative sense RNA, and it's going to have a polymerase bound, and it's going to have a lot of nucleoproteins wrapped around it. And the segments do look exactly like this, with a little crown of those 3 enzymes and a long tube that's the rest of the segment. And each nucleoprotein seems to bind to about 24 nucleotides, so that's how many nucleotides it takes to wrap around one nucleoprotein. The nucleoprotein is, like all other nucleoproteins, very positively charged, so it can bind to the very negatively charged backbone of RNA. And so that's how any binding protein would work. Positive proteins will stick to RNA or DNA. Okay, let's try this again. So here we have a little time course. And so what you're seeing is the flu virus going through different changes and ending up in different places. So at the start, we've got the flu virus, and it starts out and it gets dropped off in the cytoplasm, just like I told you. So the image on the left shows us at 2 hours. And the thing that we are watching is the PB2 protein. So remember, that's one of the 3 polymerase subunits, PB1, PB2, and PA, which stand for polymerase basic 1, basic 2, and acidic proteins. Because they have a little bit different amino acids. One's a little more acidic, one's a little more basic. Yeah, but that was just the name that people came up with, and so we're kind of stuck with it. So at 2 hours, we start out in the cytoplasm, and then the virus will get into the nucleus. And look how efficiently this happens. In the first image, you can see the cytoplasm, and you can see a dark sort of oval, like an egg shape in the center. That's the nucleus. And you can see that there's no PB2 that's got into the nucleus yet at 2 hours. But compare that to 4 hours. You basically can't see the cytoplasm. The cells would look about the same shape and have about that much cytoplasm. All you can see is the nucleus because the nucleus is lit up with all that PB2. So this is the virus going into the nucleus so it can replicate. And then at 6 hours, you can see that the virus has replicated in the nucleus and has started to export brand new copies of itself. And so between 6 hours and 8 hours, that's when we're going to make brand new virus particles, but we make those at the surface of the cell. And so you can see that the entire cell is lit up, both the nucleus and the cytoplasm, with copies of PB2. And each one of those is going to be attached to a little negative strand RNA genome that's ready to be exported out of the cell. So this is how the virus takes over and does really well. So here is the process of nuclear import. Let's have a look at this. So the the ribonucleoprotein. So this is the PB1, PB2, PA nucleoprotein, and the viral RNA, each of the segments. They have to be actively transported into the nucleus, which means that the nucleus is actually going to use energy, its precious ATP, to bring the ribonucleoprotein in. And then, the ribonucleoprotein is going to completely take over. It's a really bad idea. But here's how it happens. So look at the little diagram there. And what you can see is that the nucleoprotein itself has a small region on it called a nuclear localization signal, the NLS. And you can see that over at the left written out, NLS, nuclear localization signal. And that is going to bind to a protein called karyoferrin A. So eukaryotes, the karyo part, this means nucleus, or rather we're going to substitute this word for nucleus. I think it actually means nut. But so karyoferrin A is a protein that is routinely going to pick up things from the cytoplasm and take them into the nucleus. So this is how we manage to keep things going in the nucleus. We've got karyoferrin that's ferrying things into the nucleus. So, okay. Karyoferrin alpha is the one that's going to bind the ribonucleoprotein, and karyoferrin alpha is going to be attached to karyoferrin beta. They're going to interact in the cytoplasm, and together, karyoferrin alpha and beta and the ribonucleoprotein are going to go to the nuclear pore, and once they get there, they're going to bind these two translocation factors, things that carry proteins from one side of the nuclear pore to the other. And they're called RAN, and RAN is a GTPase, so it's actually going to use GTP and smash it in order to bring karyoferrin and the ribonucleoprotein into the nucleus, and a thing called P10, which is a small protein, which is a component of the nuclear pore complex. And so, whereas HIV had to make the Rev protein in order to hold open that nuclear pore so it could go in and out as it pleased, the influenza virus is just terrible, and so it's just going to bind to one of your proteins that was going in anyway. Think of this as somebody that hears you're going to the store and says, "Hey, can I hitch a ride? Can I come along with you?" Or think of this as somebody that basically the ribonucleoprotein is getting into the club by sort of hanging on to a group that was going to get in anyway. But there's no way it would have got in on its own. So influenza, just the worst. And so once again, that's going to be the ribonucleoprotein. The nucleoprotein specifically part of that is going to bind to karyoferrin alpha, which binds to karyoferrin beta, which binds to RAN GTPase and P10. And those are what's going to carry it right on into the club. So bam, they know the bouncer, they can get in. Yeah, that's the power of being influenza. Okay, so this is a little map of every influenza RNA segment. So there's the same bit at the 5' end and the 3' end, and this is the bit that's going to be sort of purpley brown, whatever the heck color that is. And that's the thing that says 13 nucleotides or 12 nucleotides. These are the bits that are going to fold up and they're going to attach to each other. So they're going to form the handle of the panhandle. And the big blue part is going to form the pan part, just sort of the big circle of RNA that's not bound to anything. Next to each one of those, you're going to have a segment-specific non-coding region. So it's not going to code for protein, and they're going to be different on each segment. We think the segment-specific parts might get together, and that may be what how the virus is able to count. Because it's very careful about getting exactly 8 segments, and they're one of each of its segments into each virion. It's remarkably good at doing this thing, even though, of course, there's there's really nobody to watch. Nobody's making sure, no quality control there, but the virus is able to do it for itself. Down in the 5' non-coding region, which is going to be the part that encodes the 3' region of the messenger RNA, because remember, everything's backwards. This is a negative strand RNA segment. It's like a template. When we copy that, that's going to turn the A's are going to turn the U's are going to turn into A's, and once again we're going to stutter. So just like the coronavirus polymerase stuttered on those 6 U's, the influenza virus polymerase is going to stutter on those 5 U's, and she's going to keep writing A until it gets about 200 of those A's, and that's going to be the poly A tail. So that's how it gets a nice poly A tail. Now, the really cool part of the story is how it gets a cap, because you got to have a cap and a poly A tail to make a positive sense messenger RNA, just like coronavirus did. And so remember, coronavirus, hard-working virus, brought in all 4 enzymes that it needed to build its own gulp darn cap. And it did it. It did that all for itself. Flu virus is a dirty sneak thief. And so here's what happens. And actually, yeah, okay, we need to update this. So let me tell you the updated version in words, and you're going to make the amendments on your notes. So this was correct as of 2013, but since then we've learned some differences. So yeah, pretty cool. So what we know now is that PA is actually going to snatch the cap. PB2 binds to the cap. It sort of like finds it. Think of that as sort of scouting out the scene. Yeah, it's like the criminal mastermind. PA is the pair of scissors. It's actually going to come in and it's going to find a capped messenger RNA that the cell is making, probably to try and sort of make an interferon response or something like that. And what it does is it just cuts off the cap. So PA cuts the cap. It hands it to PB2. So PB2 takes the cut off cap. Think of it as like the middleman, like the fence if you're into crime, which, yeah, I hope you're not. I think of it as like the pawn shop dealer that's going to take the stolen goods from PA, which is the little burglar, and hand it off to the PB1. PB1 just wants caps. It doesn't want any complications. Doesn't care where they came from. It just says, "Boy, I need a capped little primer that I can start with." And so PA cuts them off. PB2 hands the caps in, and bam, attaches them. And so then PB1 will extend that little piece of RNA attached to the cap, and it'll make a little messenger RNA. So the first couple of nucleotides of every influenza messenger RNA are going to be different, and they're going to have come from whatever the messenger RNA of the cell was that got decapped, basically. Yeah, boom. So that's how it works. PA does the cutting, PB2 does the fencing, does the sort of snatching, and it hands it to PB1, which just is a polymerase and boringly elongates that thing, copying its template, which is the negative strand RNA. And then once PB1 gets to the poly U's down here at the end, the 5 U's, it's going to stutter until it's made a big, long poly A tail, which is A, and there's so many of them that we don't even count them. We're just going to put a little N at the end of it. That's that little A with the N at the end. That just means that's a universal symbol for poly A tail. And the messenger RNAs are what we're going to translate to make new viral proteins. So here is the replication process, which is also quite neat. So once the virus comes in there, we're going to first run off a little positive strand messenger RNA, cap snatched. So it waits till it gets to the cell to do that, and that's fine. Then the whole polymerase complex, PB1 and PB2 and PA, are going to start again, and they're going to make another copy. And the copy they make is they're going to copy the negative sense RNA, and they're going to make a positive sense RNA. So the negative sense is the same as the one that came in with the virion. So we're going to call that the vRNA, which stands for virion RNA. This is like the one that is going to invade the cell in the first place, and it's negative sense. The complementary RNA, we're going to call the cRNA. C is for complementary, and it's complement, not complement. Because it is the reverse complement of the negative sense RNA, and it is positive sense. So yay. And then from the complementary RNA, which is an exact copy but reversed, of the negative sense RNA, we'll use that as a template to make more negative sense RNA. So it's kind of the same thing coronavirus was doing with making negative strands to make more positive strands, except that flu virus works backwards. It uses negative strands to make positive strands to make more negative strands, and that's what it packages. So yeah, same idea, just starting with the opposite thing, and still getting back to the genome that we want to send to the next cell. Okay, so once it has done this, and all this RNA copying is going to take place in the nucleus so that the virus does not have to worry about rig I or capping or anything like that. And I mean, it stole a perfectly made cap anyway, so it doesn't really have to worry about that. Then the M1 protein, the matrix protein, is going to associate with the brand new ribonucleoproteins. So we're going to send new PA, PP1, and PB2 and M1 proteins into the nucleus after they get made, after we export the messenger RNAs. So we make the messenger RNAs, they go out, we make the proteins come back into the nucleus, and then boom, they're going to form an entourage around every single RNA segment, and they're going to bring those ribonucleoproteins to the outside. And so the M1 protein is going to have a vital role to play in export. So the virus used karyoferrin to get into the nucleus, and that was nucleoprotein binding to karyoferrin, and then ran GTPase and then P10. And then we're going to use the M1 protein, and the M1 protein binds to a protein called CRIM1, which is also part of that nuclear pore complex. This is an export receptor. And so M1 binds to CRIM1, CRM1, shown right there. And that's how the influenza virus is exported from the nucleus. So I would say your job is to try and make a nice little drawing that shows how the virus is going into the nucleus and how the virus is coming out of the nucleus. That's going to be a real good test question. And I'm going to put a list of all these test questions online in the Blackboard site, so you've got them. Okay, that's been all for this segment.